Founder and producer, Professor Ahmed Mamdouh El-Sharif, Professor of Epidemiology and Infectious Disease, ex-head of Department of Veterinary Medicine, ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University, presents through this scientific veterinary channel. Technical veterinary learning. Jerusalem is our holy city. القدس لنا والأرض لنا. فلسطين حرة مستقلة من النهر إلى البحر. Malignant Cattrall Fever. Malignant Cattrall Fever has other synonyms such as malignant catarrh, malignant head catarrh, gangrenous coryza, Cattrall fever, zoonotic. Malignant Cattrall fever has fatal ending as death occur within 5 to 10 days. It is a multi-systemic disease by means that it included and cause diseases in all the systems of the body, from the nose up to the tail. It causing lymphoid proliferation and infiltration to all tissues, widespread vascular epithelial and mesothelial lesions, lesions morphologically associated with lymphoid cell. In this picture, the arrow denote to the catarrh that present in the eyes and eyes discharge and nasal discharge in the diseased cattle. Malignant Cattrall fever is a fatal, ending by 100% case fatality, lymphoproliferative disease by means that it affects on the all lymphoid tissue and vessels all over the body. Bare acute disease means that the animal may found death before you can see any symptoms. Once the clinical signs appear, no chance of recovery. It has the main form is the head and eyes form, has neurological signs, and death occur within 5 to 10 days from the beginning of the signs. This picture is clearly explain the lesions present in the head and eyes as cataract, and nasal discharge which is purulent. There is desquamation of the cutaneous parts of the nasal of this cow. Economic importance of the disease. Losses attributable to the disease can be catastrophic on rare incidents as it occurs in individual farms, as it is individual animal have been responsive to the disease. It is a neonatic disease as it is resembled in its signs to mucosal disease, blue tongue disease, and its historic resemblance to rinderpest, variable given the carrier status. Zoologic parks affected through losses of expensive imported animals. Concern for bison breeders, cattle producers, elk and deer producers. Zoonosis of malignant Cattrall fever. Malignant Cattrall fever has not been documented up till now as causing disease in humans, but caution during lambing time from taking an accidental infection. Diagnosis of the disease. First step in diagnosis is the epidemiological status of the disease throughout the world. Number 1, pathogenic risk factor. This is an electron microscope of ovine herpes virus 2, one of the causative agents of the malignant Cattrall fever. Virus classification. The pathogen here is virus. Realm Dublodanaveria. Kingdom is Hingogiveria. Phylum is Bibloviricota. Class Herpesviricetus. Order Herpes viralis. Family Orthoherpesviridae. Genus Macavirus. The both species incriminated in occurrence of this disease is alcephaline gamma herpes virus 1 or AIHV1 and oven gamma herpes virus 2 or OHV2. This picture indicates presence of the virus as green color during immunofluorescence detection of the agent within the proliferative infection of lymph nodes. Malignant Cattrall fever disease. The serotype species causing this disease are geographically dependent, by mean that according to the natural host that present. Alcephaline herpes virus 1, their natural hosts are wild beasts in Africa. Ovine herpes virus 2, their natural host is domesticated sheep all over the world. Caprine herpes virus 2, their natural hosts are goats. Alcephaline herpes virus 2, it is a non-pathogenic viral species could be detected. All of them are belonging to genus gamma herpes virus. Malignant Cattrall fever virus. Alcephaline herpes virus 1 transmitted to cattle from blue wild beast Conchetus tyrannus. Caprine herpes virus 2 closely related to ovine herpes virus 2 and has been isolated from goats. Deer herpes virus has been isolated from deers. Ovine herpes virus 2 is transmitted to cattle from sheep. Neither malignant Cattrall fever virus appears to transmit from cattle to cattle, and neither causes any disease in the principal hosts, the wild beasts and the sheep. Malignant Cattrall fever virus can be grown in eggs and the tissue culture, and this picture indicated the squamation of the cell from the baseline during tissue culture injection of malignant Cattrall fever virus to the cells. Ovine herpes virus 2 is multi-organ disease due to the viral infection has never been propagated in vitro. Complete genome sequencing have been done. It read at 135 and 621 base pair according to the viral species. Complete genome sequence of alcephaline herpes virus 1 is 130 and 608 base pair. Occurrence and prevalence of malignant Cattrall fever disease throughout the world. Alcephaline herpes virus 1 primarily present in Africa, carried by the wild beast and hartebeest, toopy, and in zoologically and wild animal parks. The arrows denoted to the animal species. Ovine herpes virus 2 is worldwide carried by domesticated and wild sheep and goats. The broad range of natural hosts for malignant Cattrall fever can be divided into two important categories. Number one, reservoir hosts, sheep, goats, and wild beasts. Number two, clinically susceptible hosts are cattle, bison, and deer. Alcifaline herpes virus 1 that cause malignant Cattrall fever is wild beast associated. It occurs in most African countries in cattle that communicable with clinically normal wild beast and hard beast. Malignant Cattrall fever is also an important disease of farm deer. Number 3, positive bison have been found in United States and Canada infected with malignant Cattrall fever. Morbidity and mortality of the disease. Morbidity and mortality of malignant Cattrall fever disease. Low morbidity in other species than cattle. In United States, outbreaks recently published researches found that they have 30 to 40%. Mortality rate is very low in water buffalo, farm deer, and fallow deer, by mean that these animals, their immune system is very strong. Mortality is 100% in cattle, white-tailed deer, axis, Bear Davis deer. The carrier species are asymptomatic, and they are wild beasts, hartebeest, tubby, sheep, and goats all over the world. Origin of transmission. Epidemiological relations of alcifaline herpes virus 1 transmission between wild beasts and other ruminant species in contact with wild daily life. Alcifaline herpes virus 1 is mainly transmitted horizontally to wild beast calves from asymptomatic reactivation of their latent infected mother to asymptomatic productive infection through the nasal secretion, by mean that mother transmitted the alcifaline herpes virus 1 to their offspring, which taken the peak at 4 months of age. Also, alcifaline herpes virus 1 transmitted from tubby, bison to the cattle species. Disease transmission. Cases mostly occur when cattle have had contact with lambing ewes and usually started at 1 to 2 months later on. Cases without apparent or recent exposure to sheep do occur but rare and is uncommon. The morbidity rate varies from area to another. Goats can also act as a source of ovine viral herpes virus infection for cattle, and rare reports of clinical disease in goats are exists. Sheep-associated malignant Cattrall fever. Sheep-associated malignant Cattrall fever occurs worldwide. Transmission of malignant Cattrall fever disease. On occasion, up to 50% of a herd may be affected in rare cases. Malignant Cattrall fever disease with both agent alcifaline herpes virus 1, ovine herpes virus 1 is almost always fatal, with rare reports of recovery in cattle. Devastating outbreaks that may be short-lived or last for several months. Malignant Cattrall fever is sporadic and presents as a single or small number of cases over a short period. Environmental risk factor of malignant catarrhal fever disease. The disease shows the greatest incidence in late winter, spring, and summer months. Copper deficiency or exposed to bracken fur might be environmental stressors that predispose the expression of the disease in cattle. Animal risk factor. Malignant catarrhal fever disease had been described over 30 species of ruminants, either wild or domesticated. In Africa, assorted wild ruminants contract the disease and suffer severe illness and high mortality rate. Similar species in zoo are commonly affected, for example, Paieri David's deer, greater coons. Among domesticated animals, all ages, races, breeds and sexes of cattle are equally susceptible, but panting, buffaloes, bison, and deer are more susceptible and suffer a more severe form of the disease than do commercial cattle. Bison are estimated to be approximately 1,000 times more susceptible to clinical malignant catarrhal fever than cattle. Disease is recorded in captive deer or farmed deer, including sika deer, rory deer, white-tailed deer, rosa deer, and red deer. Farms of deer beginning to appear in Egypt for consuming purpose. Malignant catarrhal fever is considered one of the most important diseases of farm deer. The clinical signs and the necropsy findings closely resembling those of malignant catarrhal fever in cattle, but the morbidity and the mortality can be disastrously high, resulting in heavy losses for the deer farmers. Keep on the track by following the scientific channel Technical Veterinary Learning for more accurate scientific discuss and clearance of all infectious diseases belonging to animals all over the world. Thank you for subscribing.